tutorial is designed to help you generate your flowchart for your unknown lab report. The flowchart is a diagrammatic representation of the journey you took and the biochemical tests you conducted in order to figure out the identity of your unknown microorganism. It is something you need to work on after every lab session when you get your experimental results back, as it can also help guide you to the next tests that you need to perform. In order to prepare your flowchart, you will need to keep your bacterial identification chart and colony morphology chart on hand. You will also need the results you get from the biochemical tests you perform. So make sure you keep your lab notebook up to date and record your results accurately. For the purpose of this tutorial, PowerPoint was used to construct the flowchart, but you're welcome to use other software programs you may be familiar with, such as SmartDraw or Paint. If you prefer, you may also construct your flowchart by hand. Your flowchart needs to start by listing all possible bacteria found on the bacterial identification chart. It also needs to include all the tests that you conduct, along with their possible outcomes. Under each outcome, you need to list all the bacteria that would exhibit that experimental result. Doing this will help narrow down the bacterial possibilities until you're left with one possible choice and are able to determine the identity of your unknown microorganism. Before even conducting your very first experiment for this project, you can begin your flowchart. If you have a look at the bacterial identification chart, you will see that there are two main categories of bacteria, gram-positive and gram-negative. The bacteria above the black line are gram-positive, whilst the bacteria below the black line are gram-negative. Therefore, one of the first things you will want to do is figure out whether your unknown is a gram-positive or gram-negative bacteria. In doing so, you will be able to eliminate a whole range of possibilities. The gram reaction of your unknown can be determined by performing a gram stain. There will be two possible outcomes, gram-negative or gram-positive. You will need to take all gram-negative bacteria listed under the black bar and place them into your flowchart under the heading gram-negative. You will then take all the gram-positive bacteria listed above the black bar and place them into your flowchart under the heading gram-positive. Let's pretend that you performed your gram stain and your cells stained red, indicating that your bacteria is gram-negative. You would need to indicate this on your flowchart somehow. For example, you could use different colored arrows, blue indicating the result you obtained, red indicating that those bacteria are no longer possibilities. You could also indicate this in other ways. For example, you could place an X through the arrow, indicating that your bacteria was not gram-positive, but was gram-negative. Another option would be to use a T-head, showing that these bacteria are no longer possibilities and that we will be carrying on down this lineage. For the purpose of this tutorial, we'll continue to use the blue and red arrows. Blue indicating that this was the result obtained and these bacteria are viable options, red indicating that these bacteria are no longer considered. Let's say that you then decided to confirm your gram reaction using the KOH string test. When you added KOH to your bacteria, the bacteria became sticky, once again confirming that your bacteria is gram-negative. When doing the gram stain, you will also be able to observe the shape of your bacteria under the microscope. If we look at our colony morphology chart, we know that we are dealing with these bacteria below the black line, as these bacteria are gram-negative. The bacteria above the black line are now excluded from further analysis as they are gram-positive. Looking below the black line, having a look at the shapes of our bacteria, we see that we have cocci, we have rod-shaped, and we have coccobacilli. We will need to list all of this in our flow chart. Under the rods, we will list all bacteria that are rod-shaped. Under the cocci, we will list M. catarrhalis. Under the coccobacilli, P. stewardii. Let's say that when we looked under the microscope, our bacteria was rod-shaped. So we would need to indicate that these bacteria need to be considered further, whilst these two are no longer considered. We can do this once again using our colored arrows. One of the first experiments you will set up for this project is an EMB plate. EMB plates inhibit gram-positive growth. So in this case, our gram-negative bacteria would grow. EMB plates also test fermentation. A strong fermenter would produce a metallic green color. A low to moderate fermenter would be pink or purple, whilst a non-fermenter would be colorless. If we head back to our bacterial identification chart, remember that we are dealing with bacteria below the line. We have also excluded M. catarrhalis and P. stewarti due to their shape. We now have to look at the EMB column, looking at their fermentation characteristics. F+ represents strong fermenters. F represents low to moderate fermenters, and F- represents our non-fermenters. We also need to pay special attention to S. marcescens and K. pneumoniae, because they exhibit both strong and low to moderate fermentation, so they will need to be under 2 columns in our flowchart. Moving back to our flowchart, we'll have to list those bacteria that are non-fermenters, those that are moderate fermenters, and those that are strong fermenters. Let's say that in this case, color was not produced and our bacteria were found to be non-fermenters. We would use our colored arrows to indicate that these bacteria no longer need to be considered, whilst these bacteria are the ones we will be focusing on. Let's say that moving forward, you wanted to confirm these results, so you performed a PR fermentation test, looking for fermentation of glucose, lactose, and sucrose. When you got your results back, the tubes were still red, indicating that fermentation did not occur, and confirming the fact that your unknown bacteria is a non-fermenter. You've now been able to narrow down your bacterial choices to 3 choices: P. fluorescens, P. aeruginosa, and A. faecalis. Now you'll have to head back to your identification chart. You need to compare these 3 bacteria in terms of their test results, and find a test where they differ from one another so that you can narrow down your search even further. If we have a look along these results, we see that they are all similar. Until finally we get to the gelatinase, where we see variability, and the urease, where we see variability. Either of these 2 tests could help narrow down your choices further. Let's say that you decided to do the urease test. On your flowchart, you would have to indicate that you have 2 possible outcomes. P. fluorescens and P. aeruginosa would have weak urease activity, whereas A. faecalis would have no activity. So let's indicate that on our flowchart. We indicate the biochemical test, we indicate the possible outcomes, and list the bacteria for each outcome. For the purpose of this tutorial, let's pretend that when you got your results back, your tube had a slight pink color at the top, indicating that you had weak urease activity. Once again, we indicate this on our flowchart using our colored arrows. We can now see that A. faecalis is no longer an option, and we've narrowed it down to P. fluorescens and P. aeruginosa. We then head back to our bacterial identification chart so that we can compare P. fluorescens and P. aeruginosa and try and find a test that will distinguish between the 2. However, if we look at this chart, we see that they have the same test results for all biochemical tests. So now we have to get a little bit creative. Let's look at the colony morphology chart. One of the first things we can notice is that they grow at different temperature ranges. So we could use this to help distinguish between our 2 bacteria. So the next thing we decide to do is set up a temperature assay, and we test growth at 25, 37, and 42 degrees Celsius. P. aeruginosa would grow at all 3 ranges. However, P. fluorescens would only be able to grow at 25 degrees Celsius. Let's pretend that when we get our results back, we find growth at all 3 temperature ranges, indicating that our bacteria is not P. fluorescens, but is P. aeruginosa. Even though we've narrowed down our unknown to 1 possible bacteria, we would need to confirm this result by performing additional biochemical tests. Remember that you need to perform a minimum of 10 tests for this project. For this flowchart, I listed the additional assays performed as well as the outcomes obtained. All these outcomes confirm that my unknown was in fact P. aeruginosa. When you have finished your flowchart, you will need to put it all together and then place it into your lab report. Here is the flowchart we created during this tutorial, inserted into a Word document, ready for the lab report. Be sure to give your flowchart a descriptive heading in your lab report. Let's have a look at another example of a flowchart. Once again, this flowchart starts off with the gram stain, listing all the gram-negative and gram-positive bacteria. This individual found that their bacteria were gram-negative. They set up a PEA plate and growth of their bacteria was inhibited, confirming that their bacteria was gram-negative. They then set up an EMB plate and found that their bacteria was a moderate fermenter. Looking at the PR fermentation, they found that their bacteria was able to ferment glucose, but not lactose and sucrose, thus narrowing down their options. They performed a motility test and found that their bacteria was motile. They then conducted a SIM test and found that their bacteria reduced sulfur, reduced indole, and motility was inconclusive. The citrate test was used to narrow down their options even further. Their unknown was able to use citrate as a carbon source. This identified their unknown as M. morgani. They then performed 3 confirmation tests, PAD, urea, and gelatinase. All 3 tests confirmed that their unknown was M. morgani. I hope that you found this tutorial helpful. Remember that your flowchart will look different to both examples used in this video because it will be based off the unknown that you have and the test that you perform. Good luck, you got